Hi, I'm Brian Bahnson, and I'm going to introduce you to the Bahnson Lab poster and three grad students doing projects in my lab. The first, Sean Berard, is going to tell you about a structure function study of the protein platelet activating factor acetyl hydrolase. Then Chuan Ding and Jared Miller are going to tell you about projects that they work on with the focus of understanding the role of structure and dynamics in the function of proteins using model systems, protein kinase A, and thermolysin. And I finally want to bring your attention to our lab website at the top of this background, as well as my email address, bahnson@udel.edu. Please contact me with questions you have about the research program, as well as I'd also like to welcome you to contact me in my role as chair of the Department of Chemistry and Biochemistry. And finally, I hope to meet you in the very near future.
Hi, everyone. My name is Sean. I'm a third year student in the Bahnson group. I'm going to be giving a brief overview of my research project here. But to learn a bit more about me personally, you can check out our lab walkthrough video that we'll be posting. So, as for my research project, I'm looking into the functional and structural changes that an enzyme called PAFH2 undergoes when it's exposed to oxidative stress. And PAFH2 has known antioxidant functions, namely those being the breakdown of oxidized phospholipids in intracellular membranes. Um, but the specifics of how PAFH2 binds to these membranes after exposure to oxidative stress isn't well understood. And so that's one of the questions I'm looking at answering. So based on research from past Bahnsen group members, a model of how PAFH2 is trafficked around the cell has been proposed. Uh, PAFH2 is believed to exist as a dimer under standard cellular conditions. When the cell is under oxidative stress, PAFH2 undergoes some post-translational modification and becomes a monomer that's capable of binding to a membrane. And as part of my project, I'm attempting to determine the specific residues in nature of the post-translational modification that are related to this dimer to monomer transition. So this here is a homology model of PAFH2 that was built using another enzyme that's similar to it as a template. And an interesting feature of PAFH2 in this model are the two spatially close cysteine residues in the bottom right of the model here. And one theory we have is that an intramolecular disulfide bond may be occurring between these two cysteines under oxidative stress, which could potentially destabilize the dimer. And so to investigate this idea, I'll be employing a technique called mass spectrometry-based proteomics, which is commonly used to obtain structural information for proteins. And in this type of experiment, I'll be using two cysteine-specific mass tags to bind residues that are participating in disulfides and those that aren't. And then using mass spec, I'll be able to see which cysteines are bound to either mass tag based on the increased mass of the peptides due to the bound tags. And so yeah, that was just a very small bit about my research project. Obviously, I was just scratching the surface with this presentation. So if there was anything you'd like to ask about, feel free to email me. I have my email up here on the slide. And if you have questions about the lab in general, life in Newark or anything like that, feel free to reach out to me as well. And yeah, thanks guys, and have a great day.
Hi guys, my name is Chuan Ding, and I'm a fourth year student in Dr. Banson's lab, and this is my email. I'm going to briefly go over my research, and my research focused on structural, dynamical, and functional effect of serine versus threonine phosphorylation, mainly on the subject experiment object pKa. So pKa is short for protein kinase A. It has a tetramer form. So when 4-CAMP binds to the regulatory sites, which represented in letter R, 2 catalytic subunits get released, and each of them has the capability of facilitating a phosphorylation process with the help of ATP. And it's important for activating many target proteins and enzymes via phosphorylation. So if we zoom into the pKa structure, we can see that this is the activation loop, and it's interesting that it's disordered when the phosphorylation is absent, and there is a disorder to order transition when the protein gets phosphorylated. In other words, phosphorylation activates the protein, and it's interesting that threonine is conserved at this site for over 600 million years of evolution. And it makes us wonder, why is serine not conserved there for 600 million years? Since we know that serine and threonine both has the capability of being phosphorylized. So we zoom into the structure, and this is the phosphorthreonine and the backbone. We suspect there is a potential hydrogen bound between the phosphorthreonine and the backbone. So I mimic the structure of this shape into a proline mutation. So I generated this mutation in PyMol software with a proline to mimic the structure, to mimic the constraint structure. And if we see in this picture, I just put them two together, we can see they have a rather similar pentagon structure. So we're studying this subject to see how important this restraint conformation is to the activation of pKa, catalytic subunit, by comparing the not only stability but also activity and structure of the mutations. Also, we'll be mutating 197 into serine to see what that would give us. And that's pretty much a brief introduction of my project. And again, here's my email. If you're interested, if you have any questions, feel free to shoot me an email. And thank you.
My name is Jared. I'm a first-year graduate student in the Bahnson Research Group, and my research focuses on the conformational dynamics of the enzyme thermolysin. I put my email here in this bottom left corner. If you have any questions, please feel free to contact me. This project aims to interrogate the anomalous Arrhenius behavior exhibited by numerous enzymes. On this Arrhenius plot, which is described by this equation, these red and black lines are identical at lower temperatures. However, when a certain breakpoint temperature is reached, the red line deviates from the expected black line. The enzyme no longer follows the expected Arrhenius behavior, as shown here by the red line, at temperatures higher than the breakpoint. One example enzyme this has been shown in is ribonuclease A, which demonstrated an Arrhenius breakpoint at negative 53 degrees Celsius. I am interested in exploring this Arrhenius breakpoint using the enzyme thermolysin to explore whether this behavior is the result of changes in the conformational dynamics of the enzyme. Thermolysin was the model system I chose for this research, and thermolysin is a thermostable bacterial protease. As previously mentioned, ribonuclease A had a breakpoint at around negative 53 degrees Celsius, which is not easily achievable in the laboratory setting. By studying an enzyme which functions optimally at higher temperatures, thermolysin should have a breakpoint around room temperature. Thermolysin cleaves proteins at hydrophobic amino acids. The enzyme also uses several metal ions for structural stability and catalysis. Thermolysin undergoes a conformational change during catalysis, as shown in this figure to the right. The apostate is shown in yellow, and the substrate bound state is shown in blue. The enzyme closes in on the substrate when it binds. Thermolysin was an attractive enzyme to study because it is well characterized, easy to procure, and readily forms protein crystals along with that temperature stability. The first phase of this thermolysin research has already been published by Dr. Ming Dong, who was a former graduate student of Dr. Bahnsen's. She determined that thermolysin had an Arrhenius breakpoint at around 20 to 25 degrees Celsius, which is close to room temperature. One of the proposed reasons for this deals with the shifting of amino acids in the enzyme's active site over that temperature range. If you want to read more into this, I've left the name of that paper on this slide. For my part of the project, which I've begun just a few weeks ago, I plan to use several techniques to further interrogate the conformational dynamics of thermolysin. This includes X-ray crystallography using Ringer analysis, which is what Ming used to determine the position of those amino acids over the temperature range. I plan to improve the kinetic assay that we use for thermolysin. I also plan to use NMR and molecular dynamics simulations to explore that Arrhenius breakpoint. Ming used hydrogen deuterium exchange mass spectrometry and various types of spectroscopy as well to characterize thermolysin, and those may come again in my research as well. So if you have any questions, again, please feel free to contact me at this email address, and have a great day.